Hello students, this is Dr. Lyons, and this is a mantis shrimp. A mantis shrimp is one of my favorite types of sea creatures. Why I think I like it so much is because even though it's small, it's really powerful. So this little thing that you see right here, it's kind of like a punching bag, or a punching gloves, I should say. And how this little shrimp, how it uh, uh, subdues its prey is just by beating the crap out of it. So it'll just corner a crab or something like that and just start punching it until it punches the crab into smithereens. So that's why I like these guys because they're pretty badass little marine creatures. Anyway, these things have DNA, just like all the other living things on this planet. So in chapter 10, we're going to talk about DNA, about its structure and what it is used for. So we have talked a little bit about DNA already, but we're going to go into a bit more detail here in chapter 10. In this chapter, I have broken up into two parts because it's a pretty big, pretty important chapter. So this is the part one lecture in which we are going to go into just the structure, and we're going to talk a little bit about how DNA replicates. In the second video lecture for chapter 10, we'll get into the function of DNA. So like I said, this first video lecture will be an overview, and we'll go into DNA replication. So first, very broad question, why is DNA important to us, and how would life be different without DNA? Right, so we learned that DNA was one of the four important macromolecules. We learned about how it is passed on from parent to offspring, right? So that's why we resemble our parents. And what it's actually used for inside of us is as a blueprint for making proteins. So we learned that proteins are this other type of macromolecule, and there are thousands of different proteins inside of us. And those proteins do a whole bunch of different things from allowing our muscles to work, from allowing us to break down dairy, to allowing our blood to carry oxygen in it. Proteins are used for just about everything inside of us. And if it wasn't for our DNA, we wouldn't be able to make those proteins. So why is DNA important to us? It's really important to us because without it, we wouldn't exist. Right? We just wouldn't be able to make all of those important proteins that we need in order to survive. And how would life be different without DNA? Life, as we know it, wouldn't really exist without DNA. Because DNA, as we said, it's what allows proteins to be made. And because it is what is passed on from parent to offspring, it allows for reproduction to occur. And consequently, it allows evolution to occur. So if it wasn't for DNA, there would have never been evolution on this planet. Meaning that if there was any life on this planet without DNA, it would be the very simple forms of life that we started out with before those things started to evolve. Billions of years ago until we come up to today, when life is very different from what it was back then. So life would be just completely different without DNA. So let's talk a little bit about DNA and RNA structure. First, let's talk, before we get to that, let's talk a little bit about the history of our understanding of DNA and RNA. So it was in the late 1800s that DNA was discovered. Right? Before that, we didn't even know that it was a thing, which makes sense. It's only relatively recently that we've had the ability to detect very small molecules. But it wasn't until the early 1950s that scientists realized that DNA was the hereditary material, meaning that DNA is the thing that is passed on from parent to offspring, which is kind of remarkable. The 1950s, it's not a, of course, it seems like a long time ago to probably most of you, but that's just 70 years ago, right? So probably most of you have, either parents or grandparents that were alive that were born before we knew that DNA was the hereditary material, which is remarkable to me because this is such an important facet of life on the planet, DNA being passed down from parent to offspring. And it's only relatively recently that we discovered that. So once scientists determined that DNA was the hereditary material, that set off the whole field of molecular biology, right? So looking at DNA and how it's shaped and how it works and all the various things that you're going to learn about today. So there's a really, really famous paper published in 1953 about the shape of DNA. It was published by these two guys, James Watson and Francis Crick, where they described the characteristic double helix shape of DNA. Kind of the bigger backstory to this paper is that this person, Rosalind Franklin, she took a very famous type of photograph of DNA using something known as X-ray crystallography. So she produced this image of DNA. And Watson and Crick, using some of the models that they were producing and using the image that Rosalind Franklin took, they were able to determine that it was a double helix. The really sad and really shitty thing about all this is that Watson and Crick both got a Nobel Prize for their work, but Rosalind Franklin was never awarded a prize for it. And even though her image very much helped them to this discovery. And it gets worse than that. Rosalind Franklin died not too long after this paper was published, in a large part because of the chemicals that she was using to produce this type of image. You know, back in the 50s, there was not so much in the way of OSHA or organizations for protecting workers as much as there is today. And so she got, you know, unhealthy doses of chemicals from doing this type of work. And so because she passed away, it means that she'll never get a Nobel Prize for her contribution here, because Nobel Prizes are only awarded to people that are living. They are not awarded posthumously. If, however, the Nobel Committee ever decides to start awarding rewards posthumously, I would say Rosalind Franklin is a very, very much deserving candidate for this, because she was really very much screwed out of you know, the credit that she was due for her work, and very much done so by these two guys, Watson and Crick, who by all accounts were pretty major jerkbags. Francis Crick is passed away a while ago. James Watson is still alive, and he has said some incredibly racist things. So these are bad men, and they really very much took advantage of Rosalind Franklin. So even though they did, of course, you know, contribute a major thing to science, but we do need to remember that. I think this is a good you know, reminder that, you know, scientists are people, and there's a lot of people that are jerks, and so there's a lot of scientists that just happen to be jerks. And Watson and Crick are examples of that. Okay, so to review a little bit about what you learned about DNA and RNA back in chapter 3, that was. Right, so these are nucleic acids. And in these nucleic acids are like a lot of the other organic compounds, where they're polymers that are made up of many monomers. Right, so they're polymers, and the monomer that makes them up are nucleotides. Right, so you put together a whole bunch of nucleotides, and that makes DNA or RNA. And nucleotides themselves are made of 3 different parts. So they're made of part sugar, they're made of part phosphate, and they're made of a nitrogenous base. And so you might remember that the sugar and the phosphate, they're what kind of hold together each strand of DNA or RNA, and it's the nitrogenous bases that link together 2 different strands that hold together the 2 strands in the DNA double helix. Here's a figure that you've actually seen before, right? So the blue bits here, those are the sugars. The yellow bits are the phosphates, so that's just a phosphorus connected to a couple oxygens. And the green and tan and orange, those are the nitrogenous bases. So if we kind of expand upon that, here again is the sugar, here is the phosphate, and here is one of those nitrogenous bases, thymine. Right, and so with DNA, we've got the 4 bases that are abbreviated A, C, T, and G. In the case of RNA, there's no T, but there's the letter U, which we'll talk a little bit more about that in detail in a bit. So DNA, as I was saying, it has these nitrogenous bases. So we have the A, G, T, and C, so adenine, guanine, thymine, and cytosine. And each of these 4 different bases have a slightly different structure, right? So adenine and guanine both are 2-ringed structures, whereas thymine and cytosine are single-ring structures. But then if we compare adenine and guanine, they're not identical to each other. So for instance, guanine, this carbon right here is connected to a single oxygen, whereas this carbon, that same carbon is connected to a nitrogen instead. Right, so their structure is a little bit different between the adenine and guanine and between the thymine and cytosine. So each of these 4 are unique. And because they are unique, they can be used, or a long sequence of them can be used to store information. Because they are unique, they're kind of like how the letter T and the letter A are unique from each other, or the letter, I don't know, the letter O and the letter I, they're unique from each other. And because of that, you can string together a whole bunch of these and use that whole string of letters to convey information, to convey a message. And that's the exact purpose of DNA and RNA is to convey information or messages. So DNA, of course, is double-stranded, as we've been talking about. And when you have 2 strands of DNA that are connected to each other, there are particular rules in how they match up. So when you have a nucleotide with adenine, it will only bond to a nucleotide with thymine. So A always bonds with T, and G always bonds with C. Right, so those are what are known as the base pairing rules. So each nucleotide can only bond to one other type of nucleotide. So why don't we get a little practice here? So let's say this is one strand of DNA. I want you to write down in your notes what would be the complement to it. Right, so if this is one strand of DNA, what would the other strand connected to this one be? Right, so go ahead and pause the video and see if you can figure out what those letters would be in the complement strand. Okay, so I'm assuming by now you've paused and unpaused the video. So let's take a look here. So A is always going to be paired up with T. C's are always with G. T's are with A's, and G's are with C's. All right, so pretty simple base pairing rules. You just have to remember that T and A go together and C and G go together. So now let's talk a little bit more about some of the differences between DNA and RNA. So we have talked about how DNA is double-stranded and RNA is single-stranded, but there are also some more nuanced differences between them. So for instance, the sugars that you find in the nucleotides of DNA are different from the sugars that you find in the nucleotides of RNA. Right, so DNA is known as deoxyribonucleic acid. So the ribo part, that refers to the sugar. And so this deoxy part is telling you something about that ribo sugar, about that what's known as ribose sugar. So to deoxy something, right, can you think of an element that starts with oxy? Right, obviously you're all thinking of oxygen, and if you deoxygenate something, you are removing oxygen. So what the name deoxyribonucleic acid is telling us is that it's a nucleotide where the sugar is missing oxygen or missing an oxygen or is deoxygenated in some way. Whereas RNA is just called ribonucleic acid, no deoxy part associated with it. So this is a diagram of the two sugars. So this is the deoxyribose sugar that you would find in a DNA nucleotide, and this is the ribose sugar that you would find in an RNA nucleotide. And so if you look right here and right here, you can see that on the ribose sugar, we have an oxygen and a hydrogen connected to this carbon right here. On the deoxyribose sugar, it's just a hydrogen connected to that carbon. So that very, very simple difference is why we call them two different things. Right, so this one is missing that one oxygen, so it's called deoxyribose, whereas this one has that oxygen, so we just call it ribose. And that is why we have DNA versus RNA. So that is kind of an admittedly pretty nuanced difference between them, but it is why it is where the two different names for these molecules comes from. Okay, another kind of nuanced difference, although one that will be important as we start to learn about the function of DNA and RNA, is that DNA uses the letter thymine, T, whereas RNA uses the letter U for uracil. So thymine is a nitrogenous base that DNA uses. Uracil is a nitrogenous base that RNA uses. So remember that when you are writing a strand of letters in its DNA, you're going to be using the letter T. You're not going to be using any letter U's. And if you're writing a strand that is an RNA strand, you're going to be using U, not T. So there won't be any T's present. Okay, now let's talk a little bit more about that sugar. And this applies both to the sugar of DNA and of RNA. So something that scientists do is that we tend to label things. And in the case of the sugar, the carbons that are around that sugar are labeled 1 through 5. So the carbon number 1 is this one right here, and it attaches to the nitrogenous base. Carbon number 2 is the one that connects either to the hydrogen if it's deoxyribose or the oxygen and hydrogen if it is ribose. The 3 carbon is right here, and that's going to attach to the next nucleotide that would be down here. So it would attach to the phosphate of the following nucleotide. Carbon 4 is right here, and then carbon 5 is this one here, and that connects to the phosphate of this nucleotide. So really it's carbons 1, 3, and 5 that are important. So carbon 1 attaches to the nitrogenous base, carbon 3 attaches to the next nucleotide, and carbon 5 attaches to the phosphate. And you can see I've put little apostrophes here next to the numbers. That's because we refer to these as primes. So like 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. And why we call it that is actually Amazon, believe it or not. So Amazon has a patent on your DNA. And so these things are called primes because of Amazon Prime. Did I trick you? Hopefully not. Yeah, that was false. That's not true. Amazon doesn't have a patent on your DNA. Although, they're a big evil corporation. I'm sure they would if they could. But anyway, they're just called primes for some other reason, nothing to do with Amazon. So, like I was saying, 1 prime carbon attaches to the nitrogenous base, the 5 prime carbon attaches to the phosphate, and the 3 prime carbon attaches to the nucleotide that is below it. Okay, and so why this is even important to do, why it's important to label these different carbons is because it's a useful way then to think about how 2 strands of DNA are structured. And I'll come back to that point on the next slide, the 5 prime and 3 prime ends. Okay, so when we have 2 different strands of DNA, they are arranged upside down from one another. So let's say we've got this DNA strand here, and we've got this one nucleotide right here. The last carbon in this strand of DNA going this way is the 5 prime carbon, right? So the 5 is the very last one, and then you have the phosphate. So we refer to this as the 5 prime end. Down here, this last carbon that you find right here, this would be the 3 prime carbon, because it is the last carbon that you find in this long chain, in this long sugar phosphate backbone. So DNA and RNA strands have 3 prime ends and they have 5 prime ends, right? And so, and that is based on what is the last carbon that you find in a chain going one direction or the other. So when you have 2 strands of DNA, they're attached to each other in opposite directions. So the 5 prime end of one DNA strand will connect to the 3 prime end of another DNA strand, right? You see how all these letters are upside down here? It's because this whole strand is upside down compared to this strand that is right here. And that becomes important with the nature of the machinery that is going to take one strand and make another strand from it, because it's because the machinery that does that is kind of reads the letters in the same way that you would read a sentence or a book. You read it in one direction. If you're reading like English, you would read letters from left to right. If you're reading something in Arabic, you would read the letters from right to left, right? But there's always only one direction that you go. And that directionality is because of the 5' and 3' ends. Okay, so now that you have a little bit more background on how DNA is structured, let's talk a little bit about how we replicate DNA. So first of all, let's talk about why it is important for DNA to replicate itself in the first place, right? Why do we need to replicate DNA? So let's think about this. Let's say that your parents only had, one copy of DNA inside of them. And when they had you, they obviously would give that one copy to you, meaning that they wouldn't have any DNA left. And as we know, DNA is really, really important. You have to have more than one copy of DNA inside of you. In fact, every cell in your body needs to have one copy of DNA. So it's important that DNA can replicate itself because it's needed for reproduction. It's needed to make offspring. If you can't make more DNA, then you can't have babies. You can't have offspring. So that's a really important part of DNA replication. Another reason why DNA replication is so important is for cells to be able to reproduce, for cells to be able to make more cells. So, if you go back, whatever, 20, 30 years to, to the moment at which your dad's sperm cell met up with your mom's egg cell and made you, at that very moment, you were made of one cell, and that one cell, had one copy of DNA inside of it. But now, of course, you are made up of thousands and thousands and thousands of cells. And each of those cells, they have their own complete set of DNA. So in order for all those cells inside of your body right now to have DNA, DNA replication had to take place along the way, right? So if we go back in time to when you were just one cell, essentially before that cell splits, it has to make another copy of itself. So that then when you become two cells, each of those cells have their own copy of DNA. And then when those two cells are going to split and make four cells, each of those two cells would have to make more DNA. So essentially, any time cells split and make more cells, you need to replicate the DNA. Otherwise, the cells that would be produced wouldn't have a complete complement of DNA and wouldn't be able to function. And so DNA replication doesn't just stop once you have reached your full size, once you're not getting any bigger and adding more cells to your body, because your cells also wear out, right? So even though you might not be growing anymore now, your cells wear out and have to be replaced. So the cells that are, say, in your, on the surface of your skin, those cells have to be replaced once every couple of weeks. They die and they have to be replaced. And so in order for them to be replaced, they need to make new cells. And those new cells have to have DNA inside of them. So DNA replication is actually going on inside of each and every one of us right now. Because inside of all of us right now, our cells are dying and our cells are producing more cells to replace themselves. And in order for them to be able to do that, they need to be able to replicate DNA. So DNA replication is an incredibly important process. All right, and this is more or less what I just said. When a cell reproduces, it has to duplicate its genetic information, duplicate all of its DNA, so that it can hold on to one copy for itself, and it can give one copy to a cell that comes from that cell, what's called oftentimes called a daughter cell or an offspring cell. Okay, so let's talk a little bit about how DNA replication actually happens. First, as a sort of kind of broad overview, what we essentially do is we take the two strands of DNA, we unzip them, just like you would unzip a zipper, and then we add new nucleotides onto a new strand of DNA. And so what we end up with is two different DNA molecules. And each of those DNA molecules are made of two strands. and one strand is an old strand called the parental strand, and one strand has been newly made, and that's called the daughter strand. And how DNA replication actually works is it doesn't just begin at the very beginning of your genome and go from one end to the other. It actually starts at various places, along the way. So it's a little bit different from how you might read a book. Typically when you read a book, you start at the beginning and go to the end. In the case of DNA replication, it starts at many, many different places and proceeds forward in many different places, which essentially just makes it go faster. So we start at these origins of replication where the two DNA strands are unzipped from each other. And then essentially there's this bubble that moves in directions into the left and to the right and expands outward. And as it goes, more DNA is being added onto the new daughter strand. So what you end up with at the end is two complete DNA molecules, where each DNA molecule is made of two different strands, and each strand is made part parent and part new, so part old and part new. So there's kind of two really important molecules that are involved in making this all happen. One is called DNA polymerase and one is called DNA ligase. Right, so both of these end in ace, so that should tell you about what type of macromolecule they are. Remember, we talked about lactase, right? Lactase is a protein, a type of an enzyme, which is a type of protein that breaks down lactose. So these are both types of proteins, and specifically they're both enzymes. And so what DNA polymerase does is it is the thing that actually reads the new strands, it reads the old strands of DNA and makes the new strands of DNA. So kind of like how you looked at those letters of DNA a few slides ago and wrote down a new complement strand to that one, DNA polymerase does the same thing. So it reads those letters of DNA and produces a new daughter strand from it. And kind of like how when you read a sentence, you would only read it in one direction. Like I was saying before, if you're reading a sentence in English, you would go from left to right. If you're reading a sentence in Arabic, you would go from right to left. And DNA polymerase only goes in one direction. So DNA polymerase starts reading. So it only goes from the five from the it so it the DNA that it makes is going from 5' to 3'. And because the new strand that it makes is going to be opposite from the old strand, it is reading the letters of the old strand in a 3' to 5' direction. Which I know all of that can kind of start to get confusing, all these 3's and 5's and opposites and whatnot. I guess what is really most important for you to know is that DNA polymerase only reads in one direction. It can't read in both directions. Okay, and so because when DNA is replicated, we start at a whole bunch of different places. What you end up with is a whole bunch of strands of DNA that are actually not connected to each other. And so that's where DNA ligase comes in. So DNA ligase is kind of like a glue. And what it does is it puts together the fragments of the new DNA strand. It kind of glues them together. It patches up the holes that are between the new strand of DNA. So it essentially just comes in and cleans up where DNA polymerase didn't, complete those strands. So DNA ligase is like the glue that holds the new strands together. Okay, and so that's all you need to know for now about DNA and RNA structure and about how DNA is replicated. In the next chapter, we're going to talk about the function of DNA. More specifically, we'll talk about how we start with DNA and end up with a protein.